Welcome back to another session of RNA biology, and we left in this topic, that is the transcription in prokaryotes. And transcription in any animal is the first step in the gene expression, and the gene expression, as I already told you, is a very loaded terminology, in the sense it can be influenced in multiple ways. It is not as simple as we portray or we assume. So as and when we interact with each sections, or we will deal with each sections, we will come to that topic and revisit the so-called gene expression. And remember, in the simplistic term, gene expression is the conversion of a existing genetic information into a meaningful or useful or functional stuff, let it be anything. And if you look into the stages of transcription, it is basically started with DNA dependent RNA polymerase, and it happens unidirectionally, that is 5' to 3' direction only. And the RNA polymerase walks literally on the DNA by unwinding it locally as and when required. And it also have specific upstream and downstream sequence which enables the proper defining of a gene region or proper defining of the gene's boundary. So the RNA polymerase initiates a RNA chain, and that is called RNA chain initiation. And then comes the RNA chain elongation, that is the second step in the RNA production or the mRNA transcription. And the third step is the chain termination. This production of RNA is varying, or the rate of production of an RNA from a gene vary significantly from organism to organism, and even from within one organism, gene to gene. Not all genes are produced at the same rate. Not all organisms produce a given RNA at the same rate. So it varies significantly. And if you look into E. coli RNA polymerase, E. coli is a bacterium which is living in our intestine, our body, which is actual name is Escherichia coli, and it is a well-studied model organism for various aspects of bacterial physiology, or even it has helped us in understanding how bacterial pathogens function. Pathogens cannot be used for, pathogen is those bacteria which can cause infection in you. E. coli is rather harmless bacteria which is extensively used for studying several biological processes related to prokaryotes. And it is a tetrameric core. So the E. coli RNA polymerase contain tetrameric core. What are the two alpha units? Alpha and alpha, two alpha units, and one beta and a beta dash. So these 4 protein subunits constitute the tetrameric core of the RNA polymerase in bacteria. And then if you want to have a holoenzyme, or rather a complete enzyme, this tetrameric core is the crucial part, but it is not the complete enzyme. Say for example, if someone asks you, how do you eat food? So you will say through the mouth. So the core part is the mouth, but mouth cannot exist unless you have a face, unless you have a body. Mouth cannot exist in the atmosphere, right? So the total human or total organism have a mouth attached onto their face. So a holoenzyme is a necessity for this tetrameric core to perform the actual task. So tetrameric core plus a new factor, that is sigma factor, forms the holoenzyme, or rather complete enzyme. So their molecular weight is roughly around 480,000 Dalton and can range up to 650 Dalton individual subunit, and each base pair is going up to 650 Daltons, whereas the total protein is around 480,000 Dalton. This is roughly a rough estimate for us to understand how big the protein is. And the molecular weight of proteins are usually referred in Daltons after the famous scientist Dalton. And the functions of individual subunits. What is the function of alpha subunit? It is the assembly of the tetrameric core. So it is an organizer of the enzyme. Beta, what it will do? Ribonucleoside triphosphate binding site. That means beta subunit of the RNA polymerase enables the ribonucleoside triphosphate binding. And beta dash, which is related to beta but not exactly beta. Remember, two alpha units we have which are identical. So the beta dash allows the DNA template binding region. The sigma factor allows the recognition of the region on the DNA where the transcription has to take place. Let us revisit once again. Alpha subunit allow assembly of the tetrameric core. That means all the two alpha, beta and beta dash should come together. Whereas the beta subunit allow the ribonucleoside triphosphate binding site, and beta dash, what it will do? The DNA template binding region. That means beta dash recognize, go and attach onto the template's DNA strand. And the sigma factor tells where is it. So we can call sigma factor as a supervisor. So you have a cleaning staff or a construction worker is there, they are the one who is actually doing the job. But a cleaning staff or construction worker cannot know which part of the building we should be working today. So that is governed by or directed by a supervisor. So sigma factor is somewhat like that of a supervisor. But without supervisor, function cannot happen effectively. Function will happen randomly somewhere in some way. So sigma factor is very important in deciding where or how a transcription should initiate. So sigma factor is a requirement for in vivo situation. In vivo means in real situation. If a bacterial genome is present inside a bacteria, in real time situation, sigma factor comes into picture and it decides how effectively a given gene is transcribed. But in vitro, the RNA polymerase works equally good on both the DNA strands, and sigma factor is not a necessity because entire whichever region the RNA polymerase finds in the system, you can always have a transcription taking place in vitro. In vitro means in laboratory condition, in test tube condition where you don't need to have the organism. So basically, for a RNA polymerase to function, all you need is a DNA strand and the enzyme, RNA polymerase enzyme, and the monomer, that is ribonucleoside triphosphate. They need, that is enough, and the polymerization will take place and the RNA will be formed from a given template DNA strand. So the initiation of RNA chains, that is the binding of the RNA polymerase holoenzyme. Remember, tetrameric core plus sigma factor is the holoenzyme to a promoter region onto the DNA. And then you need to have localized unwinding of the two strands by RNA polymerase to provide a single stranded template to be accessed to make a copy of the template into a complementary strand which is in the RNA form. And then the formation of the phosphodiester bonds between the first few ribonucleotides on the nascent RNA chain that is growing up. So the initiation of RNA chain contains these three steps. So we saw transcription has three phases: initiation, elongation and termination. So the initiation has got binding, unwinding and phosphodiester backbone until few bases. A typical E. coli promoter, if you are looking, this slide may look little complicated, but don't worry, it can be dissected out quite easily. Let us see, there is an arrow given here, that is the transcription, and the directionality shows left to right for our convenience. So in a cell there is no left or no right or no top or no bottom. But for our convenience on a paper, we always give left to right as the transcription, just in the similar to the convention of you write on a paper. So this is the place where the transcription is starting, and this has to be a specific region associated with a regulatory region or a promoter region, and upstream of this region around we refer to as -10 sequence. So the transcription initiation, the first base that is transcribed, we give a number +1, and the previous base, immediate left side base, we call it as -1. There is no 0. So unlike the graph, you always say +1, 0 and then -1. No, there is no 0 here because you are counting into a definitive base. So +1 or -1. So the start point is +1. Now you go backward, -1, -2, -3, -4, -5. Like that you go up to -10. You have a specific sequence that reads AT, AT, TA. So this is basically a -10 sequence, which is very important in the recognition of the DNA by the Holoenzyme. And much upstream to that, -10, then you have -11, -12, -13, -14, like that you are going 5' upstream or backwards more and more. You have a sequence AA, CT, GT, and the opposite strand is TT, GA, CA. And in between the -10 and -35, you have around 16 to 19 bases. So this can vary from promoter to promoter or gene to gene, can slightly vary. And we refer this top strand as the template strand, because which is used to make a copy of the RNA, and the non-template strand is actually the sense strand, which is what is made in the form of an RNA. So in the right hand side box, the idea is made very clear, +1 and -1, +2 and then -2. So like that the sequences are moving downstream and upstream of the transcription initiation. And the localized unwinding is shown here in the form of a widened double strand, and this double strand is showing now like a railway track parallel for convenience. Actually it is remaining like a double helical form. For convenience we have opened it up and it's like a straightforward strand. And the numbering details are given for easier understanding in text. The transcript initiation site is +1, and invariably it has to be an A or T, usually the first base. And the bases preceding the initiation site were given a minus notation, that it prefixes and are referred to as upstream sequences. When you say upstream, like in a river or in a journey, you have an upstream journey or downstream journey. So same way, for a river, you are standing in a particular location, where from the river is coming until the place where you are standing, we call it as an upstream sequence or upstream stream, upstream region. And the place where you are standing, where the river is going further, we call it as a downstream region. So same way, transcription initial point and before that we refer to as upstream, and downward is called downstream sequence. Bases following the initiation sites were always given a plus notation or plus symbol, and they prefix a plus symbol and are referred to as downstream sequence for our conventional understanding. So keep in mind, whenever someone says an upstream sequence of a gene, that is more 5' towards the sequence, and downstream means more towards the coding region of this gene. And then consensus sequence are those regions that are highly conserved, especially the -10 and -35 sequences are highly conserved. They do not allow variation. Whereas in between -35 and -10, variations in sequences are allowed. So -10 and -35 regions are conserved. -10, when you say, don't think that exactly the -10th base. When you say -35, don't think exactly the -35th base. That vicinity around 5-6 base, they usually remain conserved with a purpose, because those regions are helpful in recognizing the DNA template or the region where the gene is present by the Holoenzyme. and the recognition sequence predominantly are by the sigma factor. Sigma factor is the one which decides where the transcription must initiate. Now, the step 2 of the transcription, that is elongation. RNA initiation, transcription initiation, and now elongation. Importantly, the sigma factor needs to be released. That means sigma factor's function was to identify the location of transcription, and now it has to be released, and then and unwinding activities to continue. Initially, unwinding of the DNA happened, the transcription bubble was created, and now the open region has to be closed. So there has to be a rewinding, and then downstream region you have to continue to unwind because you want to access the template strand. So this rewinding and unwinding will continue over and over throughout the transcription bubble when it is walking. So this transcription bubble literally walk on the DNA strand in 5' to 3' direction, and as a result you end up getting a growing RNA chain, and the RNA polymerase binds both DNA template and growing RNA chain. So RNA polymerase has to make sure that the growing RNA should not fall off from this complex. If it fall off, it is a truncated RNA. So it has to catch hold of both the DNA and the RNA. So growing RNA should be held together, and the DNA template also should be held together by the polymerase. That is what you are seeing here. The transcription bubble and the RNA polymerase is in yellow color. It continues to grow, and then there will be unwinding of the DNA double helix, and see, initially it was here and now it is here, and it continues to move on top of the DNA. As a result, this green color short RNA now became longer and longer, and it will continue. Some genes are pretty long. They can go in thousands of bases, and it can take sometimes several minutes or maybe even hours to complete the transcription of one gene. Human muscular dystrophin is an example. It's a huge gene and it takes long time to complete the transcription. And now quickly look at this table which talks about the recognition sequence of sigma factor. Like we saw, tetrameric core remains constant, the same, but the sigma factor is not the same. It can vary from gene to gene. That means each sigma factor has the ability to allow the recognition of certain promoter regions, not all promoter regions. Say if E. coli have got 100 genes, one sigma factor can govern the transcription of say some 10 genes, randomly 10 genes, and the other sigma factor can govern maybe another 15 or 20 genes. Another sigma factor can govern maybe around similar number of genes. All these things together can constitute the recognition of entire genome of an E. coli. What is the difference? Say sigma 28. Basically, it is a 28 Dalton. 28 Dalton is its molecular weight, the sigma factor, and sigma 32 means 32 Daltons. Sigma 54, 54 Daltons. Sigma 70, 70 Daltons. So sigma factors are different and their molecular weight also differs. Accordingly, they have their own unique consensus sequence, minus 35 and minus 10. So sigma 28 will prefer a minus 35 consensus of TAAA, and I will not read into the details, GCCGA TAA. So this is the minus 10 sequence, and this sigma 28 governs the expression of genes important in flagellar synthesis and chemotaxis. It do not bother about other genes at all, sigma factor. Whereas tetrameric core is a must, but sigma factor is the, I told you that is the caretaker, or that is the supervisor that allows the worker to go and bind where. Sigma 32 similarly have got its own unique. If sigma 32 also had the same as that of sigma 28, then sigma 28 will take care of the sigma 30. factor. That doesn't happen. It has got a totally different sequence, CTTGAA. Similarly, it has got a different -10 sequence, and they are important in heat shock genes. That means if bacteria undergoes a high temperature region, some proteins has to be protected, some proteins has to be produced in order to protect the bacterial cell. So they are called heat shock genes. And another sigma 54 meant for nitrogen metabolism genes, sigma 70 meant for housekeeping genes. Like that, each sigma unit decides what sets of genes are produced, whereas once it is initiated, the tetrameric core remains the same in all the RNA polymerases. Now we are talking about the termination signals in E. coli. Termination is the last phase in the transcription finalization, or the termination is the stoppage of the transcription, and they are of two types: Rho-independent and Rho-dependent. So first one is Rho-dependent termination. As the name indicates, it requires a new protein factor called Rho factor, and it is given in this notation of the letter, that is Rho. And Rho-independent terminators, as the name indicates, it do not require this protein Rho for the termination to occur. Let us see what is Rho-independent terminators. And Rho-independent terminators are those which do not require the Rho factor. So let us see how the transcription is taking place. This is the DNA strand. Let us note it has a interesting sequence called inverted repeat sequence. What is inverted repeat sequence? See the sequence: TGCCGCCAG, and same thing repeated here: TGCCGCCAG from right to left. Same way this is repeated here. And this inverted repeat basically means they have the tendency to pair because they are complementary to each other. So when the transcription takes place, when this inverted repeat regions are transcribed, because this happens towards the end of transcription. See, this blue highlighted regions are transcribed. This green color is the RNA, and now it has transcribed. What happens? Once this completely transcribed, it can form a loop, because you can see a stem and a small loop region, and it allows some steric pull or some strong force that is acting onto the RNA polymerase and the transcription bubble. And subsequent to that, immediate downstream, you have got a A-rich region. If it is an A-rich region, the RNA will have a U-rich region. So there is no other base, just A. And complementary to that, in the RNA, you will have U sequence. And this sequence, which is a weak area of hydrogen binding, because bonding, that is A double bond U, A double bond U, A double bond rich region are rather weak. And AU pairing immediate downstream to this inverted repeat sequence allow a strong pulling action by the long RNA that is finished its transcription, and this secondary structure that is formed in this RNA creates a drag, a pull onto the growing RNA, because of which the transcription bubble is no more able to hold onto this RNA and it falls apart. And this is the completion of the RNA in a Rho-independent manner. So RNA transcription stops when the newly synthesized RNA molecule forms a GC-rich hairpin loop, as you can see here, and followed by a stretch of run of A's, and this creates a mechanical stress and pulls the poly-U transcript, because poly-U copied from the poly-A in the DNA. Poly-U transcript out of the active site of the RNA polymerase, and AU has got a weak double bond interaction with, so the DNA binding with the RNA is rather weak, because of which you end up getting a release of the RNA in this weak binding region. So the release of the transcript when the RNA polymerase pauses after the hairpin. After the hairpin, the RNA polymerase pauses, and this mechanical stress creates the release of the RNA transcript. So this terminates the RNA transcription. Let us see how the termination signal is made use in a Rho-dependent manner. So in Rho-dependent termination, that is non-intrinsic, and it requires a Rho utilization site or RUT site. And non-intrinsic means it depends on a external factor. Intrinsic means it is self-sufficient, like Rho-independent is a self-sufficient or an intrinsic system. And this RUT region contains specific sequence that allow the binding of this Rho protein, and it is not meant for hairpins or other secondary structure. It is sequence dependent manner. So the Rho recognition sequence, when present in the RNA, the Rho protein will come and bind onto the growing RNA molecule. So you can see here in this sequence there are long stretch of Rho binding region where the C's are 41% C's and 25% A's and 20% U's and 14% G's. So predominantly this sequence is rich in C's and then A, U and least by G's in this RNA sequence, and this allows the Rho recognition, and this termination occurs roughly around 1 to 3 bases downstream of this Rho utilization site or RUT site. So you can see here this Rho dependent termination, whenever you have this RNA molecule that has come to the Rho utilization region, the Rho protein binds onto the RNA. This again causes a mechanical stress, because an RNA, if a protein is bound onto it and the Rho moves along the RNA and following this newly growing RNA chain, and the RNA polymerase pauses at the terminator sequence, and the Rho can easily catch it up onto that RNA and it binds stably. So the Rho can start following the RNA, and whenever the RUT site comes, it will be able to bind strongly, and the Rho unwinds the DNA RNA hybrid, and this Rho protein at its extreme end, it will cause a unwinding of the DNA RNA hybrid, and which will cause the release, and that is called the termination of all components that takes place, and the Rho and the RNA gets separated. So we will continue the remaining part of the RNA biology in another lecture. I am stopping here. Thank you.